We've already discussed three other organic molecules: carbohydrates, lipids, and proteins. And now we're going to discuss another organic compound called a nucleic acid. Nucleic acids are large organic molecules that are composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus. Nucleic acids store and process information at the molecular level. Nucleic acids are either known as DNA, and DNA helps to determine things like our eye color, our size, other inherited characteristics. DNA encodes the information needed to build proteins, and we know that proteins have many functions in our body. DNA also controls the manufacture of enzymes and metabolism. RNA is a single strand of nucleic acids, and the different types of RNA work together to manufacture specific proteins. Nucleic acids consist of nucleotides that are linked together by a process called dehydration synthesis. In RNA, there's only one strand of nucleic acids, and here we see just one strand, and each one of these units would be called a nucleotide, and that's in RNA. In DNA, we see that there are two strands of nucleic acids. We have one strand here, we have another strand here. And again, these strands of nucleic acids are also comprised of nucleotides that are bound together through the process of dehydration synthesis. And then the one strand of DNA and the other strand of nucleic acid will be bound together by hydrogen bonds to form DNA. We said that nucleic acids are made of strands of nucleotides linked together. Here is a generic nucleotide that we'll take a look at to see what structures are in it. Each nucleotide has a sugar group, and the sugar group would be either deoxyribose, if it were a DNA nucleic acid, or a ribose, if it were an RNA nucleic acid. There's also a phosphate group that's attached to the sugar group, and then attached to the sugar group is a nitrogenous base. Now with DNA, the nitrogenous bases are either adenine, guanine, cytosine, or thymine. If it were an RNA molecule, the same nitrogenous bases would be used: adenine, guanine, cytosine, but in place of thymine, an RNA molecule would have uracil. RNA is single-stranded. It contains a single chain of nucleotides. Again, each nucleotide contains a phosphate group, a sugar group, which in this case is ribose, and a nitrogenous base. The nitrogenous bases, again, are adenine, guanine, uracil, and cytosine in the RNA molecule. There are three different types of RNA, which we'll discuss in Chapter 3. There's messenger RNA, there's transfer RNA, and there's ribosomal RNA. A DNA molecule consists of a pair of nucleotide chains, or a pair of nucleic acids. So we see one nucleic acid strand here, and a second nucleic acid strand here. Hydrogen bonding occurs between opposing nitrogenous bases that holds the two strands together. Adenine is always complementary to thymine. Cytosine is always complementary to guanine, and thus they are called complementary base pairs. DNA forms in a double helix where two strands of DNA twist around each other almost like a spiral staircase. I also want to mention ATP at this time. ATP is called adenosine triphosphate. Adenosine has a ribose and an adenine molecule, and attached to that are three phosphate groups, 1, 2, and 3. In between the last two phosphate groups is a high energy bond, and in between the first and second phosphate is another high energy bond. If this high energy bond could be broken, the phosphate group would break away, and the energy from that bond would be used for many different functions in the body. As we go through this semester, we'll discover all the different things that ATP is required for. Now when that phosphate group breaks away, then there will only be 2 phosphate groups, and then we would call that ADP, adenosine diphosphate.